We're going to be discussing Chapter 19, Viruses, Bacteria, and Archaeans. And this is my very favorite chapter in this course because this is my area of expertise. These are our learning objectives for today's lecture, and there is a study guide at the end of this lecture that puts all these learning objectives into question form. So we'll go over this at the end of the lecture. So I hope you guys remember from Biology 105 that different types of organisms differ greatly in details of body form and function. And this is called biodiversity. It's the sum of differences among living things. I mean, think about it. If you look inside this bayou over here, what's living in there? We have fish, we have plants, we have insects, we have organisms that you can't even see with the naked eye. And all of that together is the variation among living organisms, or the biodiversity. Now, bacteria and archaeans are single-celled, and their DNA is not contained within a nucleus. And I hope you guys remember from Biology 105 that the cells that you learned about, all the DNA was kept inside a nucleus. And I'll show you guys a picture of that in a minute. Eukaryotes are another domain of life, and those consist of protists, plants, fungi, and animals, including us. And these can be anything from a single-celled organism to multicellular, and their DNA is contained within the nucleus. So there's three domains of life. You have eukarya, which include us, and that's any organism whose DNA is contained within a nucleus. And then you have bacteria and archaeans, which are two different domains, and they're single-celled, and their DNA is not contained in the nucleus. And archaea, these guys are like tiny, tough, ancient organisms with no nucleus, and they're extreme. They live in extreme places, and we'll talk a lot more about that at the end of this lecture. And bacteria are small cells with no nucleus as well. So here's a picture of bacteria, and here's a picture of an animal cell. So these are prokaryotes, or bacteria, and these are eukaryotes. And you can see here that the DNA is just kind of floating around inside of the bacteria cell, whereas here in the animal cell, everything's very organized, everything has an organelle, and the DNA is hanging out in the nucleus. I like to use this analogy between bacteria and eukaryotes. So bacteria is like that friend or roommate's room where everything's strewn across everywhere and nothing really has a home. Like they're always losing stuff because their room's always a mess. And then eukaryotes are like the type A friend you have where everything has a home. In eukaryotes, each organelle has a very specific place inside the cell, and the DNA is always enclosed in the nucleus. So key terms. Bacterium is a member of a large group of single-celled organisms. Archaeans are a member of a group of single-celled organisms that differ from bacteria. They're extremely old. They've been around for a very long time, and they live in extreme conditions. A nucleus is a sac that encloses the DNA, and eukaryotes are organisms whose cells characteristically have a nucleus where that DNA hangs out. And the three different the three different domains of life are bacteria, archaea, and eukaryotes. And here's a great YouTube video that explains the three different domains. So within eukaryotes, remember eukaryotes are organisms that have a nucleus. Protists are the simplest eukaryotes, and we're going to learn about those next week, ranging from amoebas to giant kelps. Fungi, which are decomposers, including mushrooms, are eukaryotes. Most plants are eukaryotes, animals are eukaryotes, and we are eukaryotes. And here's a picture of a protist, a fungi, plants, and a baby. So key concepts. Life's diversity. Observable characteristics vary tremendously among organisms, and various classification systems help us keep track of the differences. Particularly, you need to know the three domains of life, bacteria, archaea, and eukaryotes, and the differences amongst them. Now, what about viruses? I didn't even mention viruses before. Simply stated, viruses are merely genetic information surrounded by a protein coat. So they actually can't be classified into any domains of life. And if you really want to see an interesting bar fight, talk to two virologists, those are people who study viruses, and ask them if they are alive or not, if viruses are alive. Viruses, literally, all they are is some sort of nucleic acid, either DNA or RNA, with protein around them, and they vary in what they look like. They are obligate intracellular parasites. So what does that mean? That means that they absolutely require a host cell to reproduce. They cannot just reproduce. Let's say you have a cold virus and you sneeze it on a desk. That virus cannot reproduce on the desk. It must get back inside your body to reproduce. It's different from bacteria and it's different from eukaryotes, and it can't be classified in any other kingdom or domain. It is its own entity. So, a virus is a non-cellular infectious agent that consists of a protein coat, and these protein coats vary in what they look like, that contain either DNA or RNA. So they are a protein coat that contains either DNA or RNA nucleic acids. What does it infect, aka who is its host, is going to completely depend on that type of virus, but viruses need a cell to replicate. So viruses are non-cellular infectious particles, a protein and nucleic acid, and replicates only inside a host cell. And these are some examples of viruses, and I'll show you guys some more in a minute. A virus is far smaller than any cell. So these are viruses. This is what an E. coli bacteria looks like, and this is the size of a red blood cell. So you can see how tiny these viruses are compared to a red blood cell. They have no ribosomes. If you remember, ribosomes are what make proteins inside the cell or any other metabolic machinery. So they don't make their own energy. What they do is they insert themselves into other cells and they hijack them, and they steal the nutrients from the cells and they steal their machinery to make more viruses. So a virus cannot replicate outside a cell, and a virus is not considered alive. Each type of virus has a structural adaptation that allows it to infect and replicate in hosts of a particular type. So, for example, viruses that are specific to dogs may not infect human cells. What kind of relationship do viruses have with their host? Very close. So, for example, if you seem to get the cold virus every year, that's because the cold virus has evolved with humans over time to do a very good job of infecting humans. Viruses infect all living things. That means there are viruses in plants, there's viruses for bacteria, and there are viruses for animal cells. If a virus infects a bacteria, it's called a bacteriophage. If it infects a plant, it's called mosaic viruses. And if it's a eukaryote, there's all sorts of different kinds of viruses that can infect eukaryotes. The structures are very based on the kind of organism the virus parasitizes. So, for example, this is a plant virus. This is a tobacco mosaic virus. And you see it looks very different from viruses like the flu virus or the adenovirus that causes the cold. Those look very different from a bacteriophage or a virus that can infect bacteria. So here's a great example of structural adaptation. This is a bacteriophage. That means this virus only infects other bacteria. Its DNA is encased in the head, which is this diamond-shaped thing. Doesn't this look like an alien or like a lunar rover landing machine? And what happens is these rod-like tails attach the virus to the host cell, and then it'll inject its DNA or RNA into the host cell. And what happens is the host cell will then make more bacteriophage. So bacteriophages are viruses that attack bacteria. The name means bacteria eaters and is commonly shortened to just phage. So if you ever hear phage therapy, that's a bacteriophage. Phage particles comprise of a head that contains the DNA packaged within a protein coat, a hollow tail by which they attach to the outside membrane of the bacteria, and then the phage DNA is injected into the bacterium, and it uses the cell's replication machinery to reproduce itself. This is a great video that shows a phage infecting a bacteria. So bacteriophages can be good and they can be bad. Right now, scientists are trying to find ways to use bacteriophage to kill antibiotic-resistant bacteria. So if you have an infection with bacteria that can't be cured with antibiotics, maybe the doctor could give you a virus that could kill that bacteria. But bacteriophages can also be really bad because they can insert toxic genes into bacteria, making that bacteria even more harmful than it was prior to being infected by the phage. So here's an example of the tobacco mosaic virus. It has a helical structure with a coat proteins arranged around RNA to form a rod, and it infects plants. It's a major problem with crops. If you guys look around right now, we are growing a ton of sugarcane that's getting harvested, and there's 4 viruses associated with sugarcane. And historically, it has been a serious disease problem in Louisiana. In fact, mosaic in Louisiana caused a near collapse of the industry in the 1920s. So farmers should be very careful about looking out for the signs of a viral infection in their crops to prevent the spread to other fields. Most animal viruses are enveloped. A membrane from a prior host encloses the virus. It's the way for the virus to escape your immune system. And all viruses contain either DNA or RNA, but they never contain both. For example, a herpes virus, so if you ever get a cold sore on your lip that's caused by herpes virus, are enveloped DNA viruses. And HIV is an enveloped RNA virus. Because a virus lacks ribosomes and other metabolic machinery, it must replicate inside a host cell. Remember, I called them obligate intracellular parasites. A virus will attach to the host cell and then enter it or insert its genetic material into the cell. The viral genes and enzymes are going to direct the host to replicate the viral genetic material and make viral proteins. And the new viral particles will self-assemble and be released. And you can see here's a virus attaching to the host cell. It gets in, it's going to release its DNA or RNA. The cell is then going to make more viruses, basically get tricked into making more viruses, and then the cell will burst, releasing those new viruses. This is an awesome video, and it talks about how the flu virus replicates inside your body. And it basically illustrates the last slide that I went over, so please watch the video. And now that we're getting close to cold and flu season, please get vaccinated for the flu. Wash your hands, and if you have to sneeze, sneeze in the crook of your elbow, not your elbow box, but your elbow, to prevent the spread of germs. So remember, what do bacteriophage infect? They infect bacteria. And bacteriophage have 2 types of replication pathways, and I will absolutely ask you on your exam about these 2 replication pathways, so please know the difference. There's the lytic pathway, in which the bacteriophage is going to multiply rapidly and new viral particles will be released by lysis. And if you don't remember what lysis is literally like the explosion of the cell. And then there's also the lysogenic pathway, and that's when the virus is going to enter a latent state and extend the viral replication cycle. So latency basically means that the virus is going to remain inside the host cell, but it's not going to kill the host cell. So here's an example of lytic bacteriophage. Remember the lytic pathway, the bacteriophage replication pathway in which a virus immediately replicates in its host and kills the host. So here we have our bacteriophage. It's going to insert its DNA into the cell. The cell is then going to make more bacteriophage, and then eventually that bacteriophage is going to get released by the lysis of the cell. So you can see the cells bursting and releasing the bacteriophage. The lysogenic pathway is a little bit different. The bacteriophage is going to insert its DNA or RNA into the host cell, and that DNA or RNA is going to become integrated into the host chromosome. And you can see here this little black piece of DNA is getting inserted into the host chromosome. And it will not kill the host immediately. It will not cause the cell to burst open. Instead, what happens is when the cell replicates, this bacteria replicates, so will that DNA. So which mode of replication would be a good therapy to kill bacteria? Hint, it's going to be the cycle that I mentioned first and is found on the left. And then which mode of replication would be a good way to insert a favorable gene into bacteria? That would be this one over here. So please know the difference between lysogenic and the lytic pathway. Here is a great video on how HIV replicates. And basically what you need to know is that the reason why we study why HIV replicates is because knowing how a virus replicates can help scientists develop therapies against those viruses. If we can prevent that virus from attaching to cells, from inserting its nucleic acid into cells, from preventing the cell from making more virus, those are all ways that we can prevent the spread of diseases like HIV. And if you weren't familiar on how you get HIV through sexual contact, Rarely nowadays you get it through pregnancy and childbirth. So we have drugs to prevent mothers from giving HIV to their offspring, injection drug use, occupational exposure, and very rarely through blood transfusion or organ transplant. And there are drugs that can prevent you from getting HIV if you are in a relationship with an HIV positive individual. If you didn't think HIV was in your backyard, you are absolutely wrong. This is from 2015, and I'm pretty sure the rates are even higher now in Louisiana. This is the HIV surveillance report, which provides national and statewide HIV and AIDS data. And what you can see here is that we have a very, very high prevalence of HIV in Louisiana, and it's not just in New Orleans, it's throughout the state. These are number of HIV diagnoses, deaths, and persons living with HIV. So you can see that persons living with HIV has increased over time, and that's because HIV is not a death sentence anymore. You can live a pretty long time with HIV as long as you're taking drugs to prolong your life. And then this is from 2015, so I'm sure the rates are higher now, but you guys can see the number of individuals in each parish per 100,000 that have HIV. So if you live in Terrebonne or Lafourche Parish, there are 200 to 300 people per 100,000 that have HIV. And a lot of people don't know that they have HIV. So if you are in a sexual relationship, I highly recommend that you use protection and get tested regularly. And our health center actually provides free HIV testing. And if you are HIV positive, please know that if you do take the drugs that are available these days, you can live well into your 50s or 60s. I mean, look at Magic Johnson. He is kicking butt, and he's been HIV positive for a very long time. So some viruses have a positive effect on human health, as when bacteriophages kill bacteria that could cause food poisoning. However, other viruses are pathogens, and pathogens are disease-causing agents. For example, the cold virus, HIV, and flu virus are all pathogens. If these viruses are transmitted to you, you will become ill. There are emerging viral diseases. Changes to viral genes as a result of a mutation or gene exchange can alter the properties of a viral disease. That's what happened with the Zika virus. So the Zika virus wasn't really that big of a deal. And then what happened was the genes changed. And in South America, it became fatal or really terrible for pregnant women carrying babies. So an emerging disease is a disease that was previously unknown, for example, the Zika virus, or is new to humans, or has recently begun spreading to a new region. So, for example, Zika in the United States is considered an emerging disease. West Nile fever, or West Nile virus, is an enveloped RNA virus that replicates in birds, and mosquitoes carry the virus from host to host. So the mosquito is called a vector for this virus. In about 1% of cases, West Nile fever attacks the nervous system and can be fatal to humans. And the West Nile fever is endemic throughout the continental United States. It's present, but at a low level. And this is a really fantastic video talking about how West Nile virus got here, which is very interesting. It was first detected in New York City, and how it's spread throughout the United States. So I use some key terms here, and I will be asking you questions about this on your exam. A vector is an animal that carries a pathogen from one host to the next. So, for example, mosquitoes carrying West Nile, or ticks carrying Lyme disease. Those are vectors. Endemic diseases are diseases that persist at a low level in a region or a population. So, for example, the cold virus is endemic to Nicholls State during November through January because there's always somebody who's got the cold virus. Another one would be the chicken pox. It's always in the environment. You guys might not remember SARS, but SARS was a really scary disease that caused a pandemic a few years ago, not a few years ago, in 2002. I was in high school then. So, sudden acute respiratory syndrome first appeared in late 2002 in China. Actually, I was in college. It quickly became an epidemic, then a pandemic, killing 774 people in 37 countries in 9 months. And it's amazing that it didn't get worse than it was. It was very scary. So an epidemic is a disease outbreak limited to one region. So like if we have an epidemic of of strep throat at Nicholls State, that is only to the Thibodeau region. A pandemic is an outbreak of disease that affects many separate regions. I always think about like worldwide or multiple countries. So let's talk about SARS. SARS is a previously unknown type of virus. And what happened was a similar virus in Chinese horseshoe bats probably evolved in other animals before infecting humans. So there was a doctor who was working in a small farming province in China in a very rural area. And that doctor treating patients attends a wedding in Hong Kong, which is a very populous city. And he stays at a very busy hotel. And what happens is, as he travels and contracts the disease, it starts to take over other countries. So, this is the ninth floor layout of the hotel that this doctor stayed in. And what you'll know is areas where the SARS virus were found, and you'll see that they are found in and around the hallway where the doctor stayed. Room K is where he ended up staying. But you'll also see in the lift hall, which is where the elevators are, that they also found SARS. So all the people staying in these rooms who take the elevator are going to end up getting infected by SARS. And they're all visiting Hong Kong, so they're going to go back to their home countries or to other countries and spread the disease. So this is a timeline. Basically what happened was that from China, from Hong Kong, the disease just spread throughout Asia and even into Canada from world travelers. And unfortunately, the majority of people who fell ill to it were hospital workers and business travelers. So what should you know so far? You should know what a virus is. You should know the 3 types of viruses we learned about and what they infect. Those would be plant viruses, eukaryotic viruses, and bacteriophages. You should know the 2 types of replication for bacteriophages. And you should know the terminology for an emerging disease, pathogen, epidemic, pandemic, and vector. Let's talk about bacteria. Bacteria are small, structurally simple cells. They're unicellular, which means they're one cell, and their other term for it is prokaryotes. And biologists describe bacteria by their shapes. So here we have a coccus, a bacillus, and a spirillum, which is spiral. And you can see the different shapes that bacteria come in. Bacteria do not have a nucleus or cytoplasmic organelles. Remember, they're like the messy room, so their stuff's just floating around everywhere. So they got some DNA, they have some ribosomes, but nothing is compartmentalized, nothing has a home like in a eukaryotic cell. A single bacterial chromosome is a circle of double-stranded DNA, and it resides in a cytoplasmic region called the nucleoid, but there is no nucleus in a bacteria. The surface structures include a cell wall, a protective capsule or slime layer, and 1 or more flagella or hair-like extensions called pili. So how small is small? Well, a eukaryotic cell is going to be the largest, then comes bacteria, and then virus. So virus is the smallest, and bacteria is somewhere between a regular human cell and the size of a virus. So bacterial chromosome is a circle of double-stranded DNA that resides in the bacterial cytoplasm. The nucleoid is just the region where the bacterial chromosome hangs out, but it's not a nucleus. And pilus are hair-like extensions from the cell wall of the bacteria and actually helps bacteria hold on to the host cell. 5 million trillion bacterial cells live on Earth. So bacteria are everywhere. Bacteria are all over your skin, they're all over your computer, they're all over your phone, they're all over everything. And that's okay. Most bacteria do not harm us. As a group, bacteria are metabolically diverse, including all 4 known modes of nutrition. So some are phototrophs, which means that they are photosynthetic. Some are chemoautotrophs, that means they get energy from inorganic sources. If you ever hear bacteria chewing up oil during an oil spill, that would be a chemotroph. You have photoheterotrophs, that means that they use energy from the sun and also eat other organisms. And then you have chemoheterotrophs, which means they break down organic compounds. Bacteria are small cells with DNA and ribosomes, but no nucleus or typical eukaryotic organelles, and they are the most abundant and metabolically diverse organisms, with autotrophs, or organisms that make up their own food, and heterotrophs, which means that they feed on others. Like all living things, bacteria need to reproduce too. Remember the whole survival of the fittest that you guys learned about? And bacteria do this in a couple different ways. One, bacteria produced by binary fission. That's a replication of a single circular chromosome and division of a parent cell into two genetically identical descendants. Literally what happens is the cell splits apart. So binary fission is a method of asexual reproduction that divides one bacterial or archaean cell into two identical descendants. So here's a picture of binary fission, and here's a great video for it. It's a method of asexual reproduction. That means that there is no exchange of DNA. What happens is one cell basically divides into two. And here again is more binary fission. Now, bacteria can also have sex. Bet you guys didn't know that. Horizontal gene transfer is the movement of genes between existing individuals, which is also known as sex, right? When you have sex, you are exchanging DNA. In conjugation, and the way I remember this is conjugal visits in prison. Well, bacteria have conjugation. One cell gives a plasmid to the other. And so I'm basically going to show this to you in a picture. So here we go. This is horizontal gene transfer. This is the transfer of genetic material or bacteria having sex. And conjugation is the mechanism of which genes exchange in which one bacteria or cell passes a plasmid to the other. So here we have bacteria 1, and bacteria 1 is going to send out a pilus, and it's going to infect this bacteria. And what's going to happen is it's going to give this bacteria a copy of its DNA, which is in plasmid form or a circular piece of DNA. And then once conjugation is over, both bacteria will then have shared this circular piece of DNA called a plasmid. So a small ring of non-chromosomal DNA is called a plasmid. So here's conjugation in bacteria again. You have this pilus. It's going to extend into the recipient, and then it's going to send over its DNA. And this pilus is actually called the sex pilus. Two other processes can also introduce new genes. A cell can take up DNA from its environment, and a virus can infect the bacteria sometimes to move genes. So remember, if a cell bursts, then the DNA will be floating around and a bacteria can pick it up, or a bacteriophage could infect that cell, and that's another way for DNA to pick up those genes. So key concepts. Bacteria have a single chromosome, and some also have one or more plasmids, and plasmids is a circular piece of DNA. They reproduce by fission, a type of asexual reproduction in which identical daughter cells are made, and cells can exchange genes by swapping plasmids and by other processes. So please know what conjugation is and transformation. And this is how antibiotics resistance occurs. So what if you don't finish all your antibiotics? So if a bacteria is hanging on the back of your throat causing strep throat, and the bacteria divide, and one undergoes a mutation or receives DNA from its environment or from another bacteria that has an antibiotic resistant gene, and you take some of your antibiotics but you don't take them all, what can happen is that bacteria can continue to divide many times, leading to antibiotic resistant bacteria inhabiting your throat. And this is a great video that basically shows you how antibiotic resistant bacteria occur. And it's a huge, huge, huge problem in medicine right now. Let's talk about bacterial diversity. Bacteria are widespread, abundant, and diverse, and they survive in all environments. Most bacteria are either harmless or beneficial, so a very small percentage of bacteria are actually pathogens. Many bacteria have essentially ecological roles, like decomposers, cycle nutrients, or form partnerships with other species. There are other bacteria out there. There are heat-loving bacteria. These are modern heat-loving bacteria may resemble early cells. They live in thermal vents similar to early Earth, and they can survive above boiling temps. For example, this bacteria called Thermus aquaticus was discovered in a volcanic spring in Yellowstone National Park. Some bacteria can make oxygen. So cyanobacteria produce oxygen during photosynthesis. It is thought that the cyanobacteria is what led to the high levels of oxygen on planet Earth way, way, way billions and billions of years ago that allowed for other organisms to live on Earth. Some bacteria will partner with fungi and form lichen. And cyanobacteria, please note, are photosynthetic oxygen-producing bacteria. Proteobacteria are the largest bacterial lineage. Those include those that are part of your normal flora, which we're going to talk about in a minute, and some very important pathogens that can make you sick. So proteobacteria are the largest group of bacteria, and you guys should know that your normal flora is bacteria that normally hangs out on or in your body. They live on your skin, in your mouth, in your intestines, lungs, vagina, etc. And they do not harm you. They're actually really good for you. If you ever eat yogurt and they say live active cultures, those are bacteria that help you digest your food. So what are the benefits of your normal flora? These are bacteria that normally reside in or on your body. One, they compete with invaders for space and nutrients. So they're always going to make sure that the bad guys don't have any space or nutrients in or on your body. They also can produce compounds which can kill other bacteria. So the bacteria on your skin right now are producing compounds that can kill pathogens. And they can lower the pH so that other bacteria can't grow. For example, ladies, in your vagina, you have bacteria there called lactobacillus that keep the pH low. Now, if your pH gets out of whack, that could lead to yeast infections or bacterial infections like bacterial vaginosis. And gentlemen, the bacteria in your gut also help you with your pH. So normal flora, the good, they produce vitamins which are unable to be produced, such as B12. They boost your immune system and they help you digest your food. Unfortunately, sometimes when normal flora get into the wrong places, they can make you sick. That's why they tell food handlers to always wash their hands after they go to the bathroom, because bacteria from your intestines, if ingested, can make you very, very sick. Bacteria can make spores. These are resistant resting stages of bacteria. They allow bacteria to survive boiling, freezing, radiation, and disinfectants. So basically, it's like an armor coat that allows bacteria to survive during poor conditions. Some bacteria can make deadly toxins. For example, bacteria that make these endospores include anthrax, tetanus, and botulism. And if you ever wonder, why do I have to get a tetanus shot all the time? Tetanus is a bacteria that makes spores. And tetanus is normally found in soil, and so it hangs out in the soil all the time. Well, once it gets inside your body, that spore will germinate, and what happens is a toxin inside of the bacteria will make all your muscles clench up, like this painting of this man here, and you basically die because your muscles cannot relax. So please go get your tetanus shot if you ever get a cut outside. Lactobacillus is a bacteria that's commonly found in yogurts and cheeses. It's also a major part of your human flora. It's found in the urinary and digestive and genital tracts, and what it does is it keeps the pH low. So if you ever look at the side of your yogurt or cheese products, you might see including active live cultures like L. acidophilus, and that's a lactobacillus. There are spring-shaped bacteria called spirochetes. Spirochetes are spiral-shaped bacteria, and they include aquatic decomposers, nitrogen fixers, and bacteria that live in cattle gut and break down cellulose. There are some pathogens that are also spiral-shaped or spirochetes, and that includes syphilis, which, if you did not know, Louisiana is #1 for neonatal syphilis cases. That means babies born with syphilis, so it's out there, so please protect yourself. And also Lyme disease that's transmitted by ticks is also a spirochete. So spirochetes are bacteria that resemble a stretched out spring. So bacteria are well studied and highly diverse. They put oxygen in the air, they supply nutrients to plants, and they break down waste and remains. Some live in or on our bodies and have beneficial effects, those are called your normal flora, and others are pathogens that cause human diseases. Finally, we're going to discuss the domain archaeans. Archaeans are superficially resemble bacteria, but comparisons of structure, function, and genetic sequences put them in a different domain. Biologists historically divided all life into two groups. You had your bacteria and you had your eukaryotes. But new evidence revealed differences in structure, organization, and gene sequences between bacteria and archaeans. They have different cell walls, they organize their DNA more like eukaryotes, and they don't share any similarity in genetic sequence with prokaryotes. So the three-domain model is now considered correct, where you have your bacteria, your eukaryotes, and your archaea. So archaea. These are the more diverse and widely distributed than previously thought. I like to think of them as the Hulk Hogan's of single-celled organisms because they like to live in extreme environments. Many archaeans are methanogens, which means they make or survive around methane. Extreme halophiles, that means they live in very highly salted areas. And extreme thermophiles, or live in very, very hot places. They can coexist with bacteria in many habitats and exchange genes with them. But think of them as really, really old and really, really extreme. So here's an example. Methanogens. These are organisms that produce methane gas as a metabolic byproduct. So they're found in places like the gut of a cow. And they're adapted to anaerobic conditions. That means there's no oxygen. So they can live with no oxygen and they can either live off of methane or make their own. Another archaean is our extreme halophiles. Extreme halophiles are organisms adapted to life in highly salty environments. So this is what you would find in the Great Salt Lakes. And extreme thermophiles are organisms adapted to life in a very high temperature environment, such as a hot spring, like exceeding 100 degrees. So archaeans were discovered relatively recently. Many are adapted to life in very hot, very salty places. Others live in low oxygen environments and make methane, and still others live beside bacteria in soils and sea, and none cause human disease that we're aware of. So, you guys need to know the difference between bacteria, archaea, and viruses. You need to know what is a virus, what are the various types of viruses, how do they look, and how do they infect their host. Can you describe lytic versus lysogenic reproduction? You need to know what is an emerging disease, and can you describe some of the most recent ones? What is a vector, and do you know an example of vector-borne disease? What is the difference between endemic disease and pandemic disease? What are the various structures of bacteria? What shapes do bacteria come in? Bacteria are metabolically diverse. Can you name and describe various ones discussed in this lecture? How do bacteria multiply? What are two different ways that bacteria exchange genetic material? What are the pros and cons of normal flora? What is the difference between a pathogen and normal flora? Why don't antibiotics work like they used to? And what are the various types of archaea and where do they live? These are your key terms.